Hello, everyone. Today, we're going to explore the ICH Q5D Guideline, the international standard for the derivation and characterization of cell substrates used in biotechnology and biological product manufacturing. This document was developed by the International Conference on Harmonization, ICH, and officially adopted in 1997 by the regulatory authorities of the US, Japan, and the EU. What makes Q5D crucial is that it aligns the expectations for how manufacturers manage their cell lines, cell banks, and all biological materials used in production. Our focus today will be understanding what this guideline means in practice for biotech companies, regulatory teams, and QA/QC professionals. The main goal of ICH Q5D is simple but powerful: to establish clear standards for how we derive, prepare, and characterize human, animal, and microbial cell lines used in producing biological products. The guideline tells us what information needs to go into a market authorization application, specifically about how your production cells were derived, tested, and controlled. In short, Q5D is about traceability, reproducibility, and safety, making sure every lot starts from a clean, characterized, and well-documented cell substrate. This guideline was developed because, historically, biologics manufacturing faced real problems. We had incidents where cell-derived products carried contaminants, viruses, mycoplasma, or even unwanted genetic material. Recombinant DNA products introduced additional risks, since genetic constructs can behave unpredictably if not properly characterized. Q5D ensures that we no longer treat the cell line as a black box. Instead, we establish it as a controlled, well-understood system that directly determines the quality and safety of the drug substance produced. Now, let's clarify what Q5D covers. It applies to all banked cell substrates, human, animal, or microbial, that are used to produce biotech or biological products. It includes both continuous lines, which are immortal, and diploid cells, which have a finite lifespan. It also applies to bacteria, fungi, and yeast. However, the guideline doesn't apply to primary cell cultures. That's covered in Appendix 1, and it doesn't apply to reagents for in vitro diagnostic use. When you submit data under Q5D, regulators expect a complete picture of the origin and history of the cells. That means identifying the original donor or species, the tissue or organ of origin, health and physiological conditions, any manipulations, such as transfection, fusion, cloning, or selection, and details about the culture medium, especially if it included animal-derived materials like serum or enzymes. This information helps regulators assess contamination risks and overall product safety. Remember, gaps in documentation aren't always fatal, but they mean you'll need much stronger testing to compensate. One of the core concepts in Q5D is the two-tier cell banking system. We have Master Cell Bank, or MCB, is the main, fully characterized stock derived from the initial clone. Working Cell Bank, or WCB, is produced from the MCB and used directly for manufacturing. This structure ensures consistency between batches and generations. Manufacturers should describe how often new banks are created, the expected number of production runs from each bank, and the criteria for bank qualification. Importantly, if culture changes don't affect product quality, you don't need to re-clone or re-bank. Flexibility is allowed, but the risk must be justified. This section of the guideline focuses on the how you bank, store, and safeguard your cells. You must describe the preparation process, media, cryoprotectants, storage vessels, conditions of freezing and storage, typically in liquid nitrogen, measures to prevent microbial contamination or cross-contamination. Equally important, your business continuity plan. You must have redundancy, multiple freezers, backup power, automated liquid nitrogen filling, and sometimes even off-site storage of your MCB or WCB. The goal is simple: ensure that your production source is always available and never compromised. This is the heart of Q5D, the testing program that defines the quality of your cell banks. Testing focuses on three key attributes. Identity by confirming the cell is what it claims to be. Purity by ensuring it's free from microbial or cellular contaminants. Stability by proving the cell line consistently produces the intended product. Testing frequency. Perform full identity and purity tests on each MCB. Perform purity and limited identity tests on each WCB. Conduct stability testing once per product during development or registration. Let's dive deeper into the tests. Formetazoan cells, morphology, isoenzyme analysis, cytogenetics, or DNA fingerprinting confirms species of origin. The presence of unique markers or product expression also supports identity. For microbial cells, selective media and biological tests confirm strain and plasmid identity. Purity tests screen for bioburden, mycoplasma, and viruses. Follow pharmacopoeial methods, USP, pH, UR, JP. Check for cross-contamination from other cell lines. Remember, even small contamination events can derail regulatory approval. Purity testing is stability testing ensures the cell's substrate remains consistent across its lifespan and storage. Two aspects. During cultivation, test cells from early and late passages to ensure they produce the same product and retain genetic integrity. During storage, demonstrate that frozen cells maintain viability and performance after thawing. For recombinant DNA systems, you'll also confirm that the inserted genetic sequence hasn't changed over time. Karyoology assesses chromosome integrity, and tumorigenicity checks whether cells form tumors in animals. For well-characterized cell lines like MRC5 or WI38, additional testing isn't needed. Their behavior is well established. But for new or untested diploid lines, you must show diploid karyology confirmation, no tumorigenic potential. If your final product contains no viable cells, for example, purified monoclonal antibodies, karyology and tumorigenicity testing are generally unnecessary. Appendix 1 deals with primary cells, such as those used in certain viral vaccines. Primary cells can't be banked, so they must be tightly controlled from the source. Key points. Donor animals must be healthy, disease-free, and preferably from specific pathogen-free colonies. Materials of animal origin used for culturing must be tested for contaminants. Testing for adventitious viruses must be done during and after production. Because primary cells are short-lived, you can't test everything in advance, so concurrent testing becomes essential. Compliance with ICH Q5D is not optional. It's the foundation of regulatory review in biologics manufacturing. Your company's ability to demonstrate control of its cell substrates is directly tied to your approval timeline. Moreover, this guideline underpins GMP inspections. Auditors often trace back production issues to weaknesses in cell banking or documentation. Strong adherence builds confidence and reduces global regulatory friction. Let's sum it up. White-heavy checkmark cell banking ensures manufacturing consistency. White-heavy checkmark full documentation of the cell's history is mandatory. White-heavy checkmark regular requalification guarantees long-term quality. White-heavy checkmark harmonization simplifies global submissions. In simple terms, if your cells are controlled, your product is controlled. To close, consider these questions for your own organizations. How thoroughly is your cell line history documented? What's your strategy for preventing cross-contamination between banks? Are your redundancy systems sufficient to survive a freezer failure? And finally, how can digital QA tools streamline your cell bank traceability? These questions are at the core of operational excellence under ICH Q5D. And answers for these questions are, for how thoroughly is your cell line history documented? Answer in practice is, maintain auditable electronic records from the donor source to the master cell bank, MCB, covering every experimental step and test performed. This enables both scientific traceability and regulatory defensibility. What's your strategy for preventing cross-contamination between banks. Answer and practices. Create procedural segregation between different cell lines, enforce controlled access, and perform identity confirmation via DNA, fingerprinting, or isoenzyme profiling on both the MCB and WCB. Are your redundancy systems sufficient to survive a freezer failure? Answer in practice is a mature redundancy plan mirrors disaster recovery in IT. Distribute risk physically and operationally. Leading biologics facilities maintain a mirrored bank at a secondary site under identical validation. How can digital QA tools streamline your cell bank traceability? Answer in practice is invest in digital laboratory information management systems, LIMS, and quality management systems, QMS, integrated with cloud validation frameworks. This turns compliance from a reactive exercise into a proactive data-driven assurance model.